The future changed by magic gene scissors. Every living thing, including humans, has its own unique genetic information. DNA is the substance that carries genetic information. DNA contains 4 types of bases: adenine, guanine, cytosine, and thymine. These bases do not pair randomly. Adenine pairs with thymine, and cytosine pairs with guanine. Like puzzle pieces, only matching pairs fit together. This is called complementary base pairing. Then why does the order of the 4 bases matter? It is because the sequence of the bases determines an organism's genetic information. If the DNA base sequence changes, or part of a chromosome is damaged by ultraviolet light or other factors, new traits that differ from those of the parents can appear. This phenomenon is called a mutation. Examples include Down syndrome, seedless watermelons, albino tigers, and 4-leaf clovers. In nature, genetic information can change by chance in this way. Then, is it possible for people to create these changes in the direction they want? The technology that came from this idea is the gene scissors. When hearing the term gene scissors, it is easy to imagine cutting DNA with a pair of scissors. But no actual scissors are involved. Instead, scientists use DNA cutting enzymes that find and cut the desired part of the DNA. They are called gene scissors because they can cut a specific part like a pair of scissors. If DNA is cut, does the gene change right away? No, it does not. Cells can reconnect broken DNA. When DNA breaks, the cell tries to repair it on its own. It is like finding a torn piece of paper and taping it back together. Then, what if we create the change we want at the moment the DNA reconnects? That is gene editing. Gene editing mainly uses 2 methods. One removes the function of a specific gene, and the other replaces an existing gene with a new one. If some DNA is lost when the DNA reconnects after being cut, that gene can no longer function properly. On the other hand, if a piece of DNA from another gene is inserted, the new DNA takes its place and replaces the original gene. It is similar to deleting an unnecessary sentence in a book or replacing it with a new one. How can we accurately find the DNA we want? This is where CRISPR-Cas9 comes in. It is one of the most widely used gene editing tools today. Scientists originally knew CRISPR-Cas9 as part of the immune system that protects bacteria and archaea. However, in 2013, researchers discovered that it could be used for gene editing, bringing a major change to the field of life science. CRISPR-Cas9 has 2 key components: guide RNA and Cas9. Guide RNA finds the target DNA. It works like a navigation system. Enter the destination, and it leads the way. When the guide RNA says, "This is the target," Cas9 cuts the DNA at that location. One finds the target, and the other cuts it. So, where do scientists actually use gene scissors? They use them to create animals or cells for disease research. With gene scissors, scientists can create laboratory animals with diseases such as osteoporosis, muscular dystrophy, and Alzheimer's disease. By studying these animals, they learn how diseases develop and develop new treatments. Can gene scissors be used directly to treat human diseases? One area of medicine under active research is transplanting animal organs into humans. What if organs from animals such as pigs could be transplanted into humans? It could greatly help solve the shortage of donor organs. Gene scissors can modify animal genes to reduce problems that may arise after transplantation into humans. However, many issues, including safety, still need to be solved before animal organs can be transplanted into humans. Gene scissors can be used not only in animals, but also in plants. even the bread we eat has a connection to gene scissors. When bread is baked at high temperatures, a substance called acrylamide can form in the burnt parts. This happens because an amino acid called asparagine in wheat changes when exposed to high heat. Then, what if the amount of asparagine in wheat could be reduced? Researchers are using gene editing to develop wheat with less asparagine than ordinary wheat. Gene scissors can also be used in food production. Researchers are developing potatoes that do not produce the toxic substance solanine, as well as larger cattle and fish by disabling genes that limit muscle growth. Gene scissors can even change the food we eat. This raises an important question. Is it acceptable to change the genes of living things as people wish? Mutations also occur naturally all the time. Should naturally occurring mutations and organisms created with gene scissors be viewed in the same way? There is another question. If gene editing can help treat diseases or increase food production, how far should it be allowed? Gene scissors are an amazing technology that can greatly help people, but they may also cause unexpected changes. In particular, we need to carefully examine what effects gene-edited organisms may have if they spread into the natural environment. Gene scissors is a technology that can directly change DNA, the blueprint of life. In the future, whether this technology becomes a powerful tool for treating diseases and solving food shortages, or creates new problems, depends on how we use it. Deciding the future of gene editing is not only the responsibility of scientists. It should be a concern for all of us.